Hello, everyone, and welcome. Today, we're going to explore some fascinating topics in biology, speciation and population ecology. This is the first slide of our presentation, so I'll keep it brief. We'll be covering how new species form and how populations interact within ecosystems. I'm excited to take you through these concepts, so let's get started. Let's explore some fundamental concepts about species and how they form. When we talk about micro versus macroevolution, we're referring to the scale at which evolutionary changes occur. Microevolution involves small changes within a species, like variations in color or size, while macroevolution looks at larger shifts that can lead to the emergence of new species over time. Next, we have clines, which are gradual changes in traits across a geographical area. This illustrates how populations can vary smoothly rather than suddenly, supporting the idea that speciation can be a continuous process. Speaking of speciation, there are different patterns, such as anagenesis and cladogenesis. Anagenesis involves a single species gradually transforming into a new form, while cladogenesis is when one species splits into two or more distinct species, creating a branching pattern. When it comes to modes of speciation, allopatric speciation occurs when populations are geographically isolated, leading to divergence. In contrast, sympatric speciation happens without physical barriers, often through ecological or behavioral differences within the same area. Now, the tempo of speciation can be gradualistic, meaning changes occur slowly over time, or punctuated, where new species appear relatively suddenly after long periods of stability. Reproductive isolating mechanisms are crucial because they prevent gene flow between emerging species. These can be prezygotic, like differences in mating behaviors, or postzygotic, such as hybrid sterility. Sometimes, after populations come into contact again, what we call secondary contact, reinforcement can occur. This process strengthens reproductive barriers to prevent the formation of less fit hybrids. Understanding species concepts is key, as different definitions, like biological, morphological, or phylogenetic, highlight various ways scientists identify and classify species. Finally, areas called hybrid zones, where two species interbreed, often show introgression, which is the movement of genes across species boundaries. This gene flow can influence the evolutionary trajectory of the involved species. All these concepts together help us understand the complex and dynamic nature of how new species arise and evolve over time, fitting into the broader theme of evolutionary biology. Today, we're going to explore two main modes of speciation, which are fundamental to understanding how new species form. First, there's allopatric speciation, which occurs when populations are geographically separated. This separation prevents gene flow between groups, allowing them to diverge over time due to natural selection, genetic drift, and mutation. Essentially, physical barriers like mountains, rivers, or distance play a key role here. The second mode is sympatric speciation, where new species arise within the same geographic area. This can happen through mechanisms like reproductive isolation caused by behavioral changes, ecological differences, or genetic mutations that lead to incompatibilities. Both modes are crucial because they highlight different pathways evolution can take, shaping the incredible diversity of life we see today. Understanding these modes helps us grasp the complex processes driving speciation across various environments and contexts. Today, I want to talk about isolation and divergence in allopatry. Allopatric speciation happens when populations become physically separated from each other. This separation can occur in a few different ways. For example, a population might colonize a new habitat, establishing itself somewhere isolated from the original group. Alternatively, a new physical barrier, like a mountain range or a river, can split a widespread population into two or more isolated groups. These barriers prevent gene flow between the groups, which is key to speciation. Over time, these isolated populations can diverge genetically, leading to the formation of new species. This process highlights how physical separation is a major driver of biodiversity, especially in the context of evolution and adaptation. It connects to the overall theme by illustrating how geographic barriers can lead to the development of distinct species over time. Today, let's explore the concept of isolation and divergence in sympatry. Sympatric speciation happens when populations that live in the same geographic area start diverging into different species. Even though they share the same space and gene flow is ongoing, natural selection can still drive divergence. This means that certain traits become favored in different subgroups, leading to reproductive isolation over time. Additionally, polyploidy, which is a type of mutation where the number of chromosomes doubles, can also lead to sympatric speciation. This often happens in plants and can create instant reproductive barriers. Understanding how speciation can occur despite gene flow highlights the complexity of evolutionary processes. It shows that divergence isn't always about geographic separation, but can result from these other mechanisms that promote divergence even when populations are in close contact. Let's take a closer look at this model for adaptive radiation on island chains. Essentially, this diagram illustrates how a single ancestral species can give rise to multiple new species as they adapt to different environments across a series of islands. The key idea here is that islands act as natural laboratories for evolution, providing isolated habitats where species can diversify without much competition from their relatives. What's fascinating is how this process is driven by natural selection. Different islands impose different selective pressures, leading to divergence in traits like beak shape, size, or feeding behaviors. This diversification results in a wide variety of species, each uniquely adapted to their specific island environment. This model connects to the overall theme of our presentation by highlighting how geographic isolation and environmental differences fuel speciation, especially in island settings. It's a perfect example of how evolution can produce incredible diversity from a common ancestor, showcasing the power of adaptive radiation in shaping life on Earth. So let's explore whether adaptive radiation only happens in allopatry. When we talk about adaptive radiation, one classic example is the cichlid fishes in the eastern African Rift Valley lakes, like Lake Malawi, Lake Tanganyika, and Lake Victoria. These lakes are famous for their incredible diversity of cichlid species that have evolved rapidly. Now, traditionally, we think of adaptive radiation occurring when populations are geographically separated, or allopatric. But in the case of these cichlids, there's some fascinating evidence suggesting that adaptive radiation might also occur in sympatry, meaning within the same geographic area. There are hints that some of this speciation could be associated with sympatric processes, where new species evolve while living in the same environment, possibly driven by factors like ecological niche differentiation. However, it's also possible that what looks like sympatric speciation might actually be microallopatric, where very small, localized populations become isolated initially, then diverge. This raises an important point for our understanding of adaptive radiation. It's not always a simple case of populations being separated by physical barriers. Sometimes, the processes are more nuanced, involving subtle population structures within the same habitat. Connecting this to the overall theme, it highlights that adaptive radiation is a flexible and complex process, capable of occurring under different geographic and ecological contexts, not just in strict allopatry. It broadens our perspective on how biodiversity can evolve rapidly in various settings. Let's dive into the fascinating world of species and speciation. First, we have microevolution versus macroevolution. Microevolution involves small genetic changes within a species over relatively short periods, while macroevolution refers to larger, more significant changes that can lead to the emergence of new species over longer timescales. Next, we encounter clines, which are gradual changes in a trait or in the frequency of a gene across a geographical area. These clines can tell us a lot about how populations adapt to their environments gradually. When we talk about speciation patterns, two main types come up: anagenesis and cladogenesis. Anagenesis is when a single species gradually transforms into a new one without splitting, whereas cladogenesis involves a splitting event, leading to two or more distinct species. Moving on to modes of speciation, allopatric speciation occurs when populations are geographically separated, preventing gene flow and leading to divergence. In contrast, sympatric speciation happens without physical separation, often through ecological or behavioral differences within the same area. The tempo of speciation can be gradualistic, meaning it happens slowly and steadily over time, or punctuated, where long periods of stability are interrupted by rapid bursts of change. Reproductive isolating mechanisms are crucial. They prevent different species from interbreeding and producing viable offspring, maintaining species boundaries. These mechanisms can be prezygotic, like differences in mating behaviors, or postzygotic, such as hybrid sterility. Sometimes, after species diverge, they come into secondary contact, which can lead to reinforcement, where natural selection strengthens reproductive barriers to prevent hybridization. Speaking of species, there are various concepts to define what a species actually is: biological, morphological, ecological, and phylogenetic, each providing different perspectives. Finally, in areas where different species come into contact, hybrid zones can form. These zones are regions where interbreeding occurs, leading to introgression, the movement of genes from one species into another, which can have significant evolutionary implications. All these concepts are interconnected and help us understand the complex processes driving the diversity of life we see around us. Let's take a closer look at speciation tempo. When we talk about tempo, we're referring to the rate or speed at which speciation occurs. Some species diverge very rapidly, sometimes within just a few generations, especially in response to strong environmental pressures or reproductive barriers. Others might take millions of years to fully diverge. Understanding the tempo is crucial because it helps us grasp how quickly biodiversity can expand or contract. Fast speciation can lead to bursts of new species in a relatively short period, which is often seen in adaptive radiations. Conversely, slow speciation might be associated with more gradual genetic changes accumulating over long timescales. This concept ties into the broader theme of evolutionary processes, showing us that speciation isn't a uniform process. It varies depending on ecological, genetic, and geographic factors. Recognizing these differences in tempo allows us to better understand the dynamics of biodiversity and how ecosystems evolve over time. Let's dive into species and speciation. First, we have micro versus macroevolution. Microevolution refers to small genetic changes within a population over time, while macroevolution involves larger, more significant changes that lead to the emergence of new species. Next, we encounter clines, which are gradual changes in traits across geographic areas, illustrating how populations adapt to different environments. When we talk about speciation patterns, two main types emerge: anagenesis, where a single species gradually transforms into a new one, and cladogenesis, where a lineage splits into two or more distinct species. Now, regarding modes of speciation, allopatric speciation occurs when populations are geographically separated, leading to divergence. In contrast, sympatric speciation happens within the same area, often through ecological or behavioral differences. Considering the tempo of speciation, we have gradualism, where changes occur slowly over time, and punctuated equilibrium, which suggests rapid bursts of change followed by periods of stability. Reproductive isolating mechanisms are crucial. They prevent gene flow between species, maintaining their distinct identities. These can be prezygotic, like behavioral differences, or postzygotic, such as hybrid sterility. Secondary contact and reinforcement come into play when previously isolated species come back into contact. Reinforcement strengthens reproductive barriers to prevent hybridization. Moving on, understanding different species concepts helps us define what a species truly is, depending on criteria like reproductive isolation, morphological differences, or genetic similarity. Hybrid zones are regions where two species interbreed, creating hybrids and introgression refers to the incorporation of genes from one species into another through hybridization. All these concepts highlight the complexity of how new species form and evolve, which ties back to our overall theme of understanding biodiversity and evolutionary processes. Reproductive isolating mechanisms are really fascinating because they help maintain the boundaries between different species, even after those species come into contact again following a period of separation and divergence. Essentially, these mechanisms act as barriers to prevent gene flow. There are two main types. First, we have prezygotic mechanisms, which are like gatekeepers. They make it very unlikely that mating will even occur between different species. These can include things like differences in mating behaviors, timing, or even incompatible reproductive organs. Because of these barriers, interbreeding becomes highly unlikely. Then we have postzygotic mechanisms. These come into play after fertilization has occurred. They often result in hybrids that are sterile, like mules, or hybrids that fail to develop properly, which prevents the hybrids from passing on their genes successfully. Both of these mechanisms are crucial because they help maintain the distinct identities of species, even when they come into contact again. They support the broader theme of speciation and how new species remain distinct over time. Let's take a moment to review a summary of reproductive barriers between closely related species. These barriers are crucial because they help maintain species boundaries despite potential overlaps in their habitats or ranges. Essentially, reproductive barriers are mechanisms that prevent different species from interbreeding successfully. They can be classified into prezygotic barriers, which occur before fertilization, and postzygotic barriers, which happen after fertilization. Prezygotic barriers include things like differences in mating behaviors, incompatible reproductive structures, or timing that doesn't align for breeding. Postzygotic barriers involve issues like hybrid inviability, where the offspring don't develop properly, or hybrid sterility, as seen in mules, which are sterile hybrids of horses and donkeys. Understanding these barriers highlights how species remain distinct and how evolutionary processes reinforce these differences over time. This summary ties into the overall theme of how reproductive isolation is a key driver in speciation, helping us understand the diversity of life we see today. Let's explore the fascinating topic of species and speciation. First, we'll look at micro versus macroevolution. Microevolution refers to small genetic changes within a species over shorter timescales, while macroevolution involves larger, long-term changes that can lead to the emergence of new species. These processes are interconnected and form the foundation of how we understand biodiversity. Next, we have clines, which are gradual changes in traits across geographic areas. Clines help us see how populations adapt gradually to their environments, blurring the lines between distinct species sometimes. Moving on, we encounter different patterns of speciation, anagenesis and cladogenesis. Anagenesis involves a single species gradually transforming into a new form, whereas cladogenesis is when a species splits into two or more distinct species, leading to increased diversity. Now, let's talk about speciation modes. Allopatric speciation occurs when populations are geographically separated, preventing gene flow and allowing them to diverge independently. Sympatric speciation, on the other hand, happens within the same area, often due to differences in ecology or behavior that lead to reproductive isolation despite ongoing gene flow. Regarding the tempo of speciation, we have gradualistic and punctuated models. Gradualistic suggests that speciation occurs slowly and steadily over time, while punctuated models propose that species remain relatively unchanged for long periods, then undergo rapid changes during brief events. Reproductive isolating mechanisms are key to maintaining species boundaries. They prevent gene flow between populations and can be prezygotic, like differences in mating behaviors, or postzygotic, such as hybrid sterility. When isolated populations come into secondary contact, reinforcement can occur, strengthening reproductive barriers to prevent maladaptive hybridization. We also need to consider species concepts, which are frameworks for defining what a species is. Different concepts emphasize reproductive isolation, genetic distinctness, or ecological roles, helping us understand and classify the diversity of life. Finally, hybrid zones and introgression are fascinating areas. Hybrid zones are regions where different species meet and interbreed, creating hybrids. Introgression refers to the gene flow from one species into another through hybridization, which can introduce new genetic variation and influence evolutionary trajectories. All these concepts tie together to give us a comprehensive understanding of how species originate, evolve, and interact within the grand tapestry of life. Let's talk about secondary contact and reinforcement. Secondary contact occurs when two populations that have been isolated from each other come back into contact after a period of divergence. During this time, they've been evolving separately, developing genetic differences. What happens next really depends on whether reproductive isolating mechanisms have developed during that isolation. If they haven't, there's a chance that the populations will interbreed freely, which could lead to gene flow and potentially merging of the populations. However, if reproductive barriers are already in place, the contact might not result in hybridization. Instead, we see a process called reinforcement. Reinforcement is about maintaining the distinctness of each population. It involves mechanisms that prevent hybridization, often because hybrids tend to have lower fitness. For example, hybrids might be less viable or less successful at reproducing, which naturally discourages interbreeding. This process helps keep the populations genetically separate, even when they come back into contact. So, in the context of evolution, reinforcement plays a crucial role in maintaining species boundaries and preventing the blending of distinct populations, which ties back to the broader theme of how species evolve and stay separate over time. Today, I want to introduce you to Ensatina eschscholtzi, which is a fascinating example of a ring species. A ring species is a situation where a series of populations of a species are distributed around a geographic barrier, like a mountain range or a valley, and they gradually change as they move along this ring. In the case of Ensatina, you can see how different populations exhibit slight variations in their physical traits as they spread around the Sierra Nevada mountains. What's really interesting is that the populations at the ends of this ring are actually so different that they can no longer interbreed, even though they are connected by the intermediate populations. This example beautifully illustrates the concept of how species can evolve gradually over geographic space, leading to reproductive isolation in some regions. It's a perfect demonstration of the process of speciation in action, fitting into our overall theme of how diversity arises through evolutionary processes. Let's dive into species and speciation. First, we have micro versus macroevolution. Microevolution involves small changes within a species, like gene frequency shifts, while macroevolution refers to larger, more significant changes that can lead to the emergence of new species. Next, clines are gradual changes in traits across geographic areas, which can tell us a lot about how populations adapt over space. When we talk about speciation patterns, two key processes come to mind: anagenesis and cladogenesis. Anagenesis is a gradual transformation of one species into another, whereas cladogenesis involves the splitting or branching of a lineage into multiple species. Moving on, there are different modes of speciation: allopatric and sympatric. Allopatric speciation occurs when populations become geographically isolated, leading to divergence, while sympatric speciation happens within the same area, often through ecological or behavioral differences. Now, considering the tempo or pace of speciation, we see it as either gradualistic, slow and steady, or punctuated, with rapid changes followed by long periods of stability. Reproductive isolating mechanisms are crucial because they prevent gene flow between diverging populations, maintaining species boundaries. Sometimes, when two populations come back into contact after being separated, secondary contact, reinforcement, can occur. Reinforcement strengthens reproductive barriers to prevent unfit hybrids. Finally, understanding species concepts helps us define what a species really is. And when hybrid zones form, regions where different species meet and interbreed, we see introgression, or gene flow between species, happening. All these concepts are interconnected and help us understand the complex process of how new species arise and evolve over time. Let's take a closer look at Table 23.1. This table provides a clear overview of the different types of financial statements that organizations use. As you can see, it outlines the purpose of each statement, the key users, and the main content they include. For example, the balance sheet, also known as the Statement of Financial Position, shows what the organization owns and owes at a specific point in time. It's essential for understanding the company's financial health. Then we have the income statement, or the statement of earnings, which captures the organization's revenues and expenses over a period. This helps stakeholders assess profitability and operational performance. The statement of cash flows, on the other hand, details how cash moves in and out of the organization, providing insights into liquidity and cash management. Finally, the statement of changes in equity explains shifts in the owner's interest, including profits retained or distributed. Connecting these points to our overall theme, these statements work together to give us a comprehensive picture of the organization's financial position, performance, and cash management. They are fundamental tools for decision-making, whether you're an investor, manager, or regulator. Understanding how each piece fits into the bigger picture is crucial for informed financial analysis. Let's dive into the fascinating world of species and speciation. First, we have micro versus macroevolution. Microevolution refers to small genetic changes within a population, like slight variations in color or size, while macroevolution involves larger, more significant changes that can lead to the emergence of new species over longer periods. Next, we encounter clines, which are gradual changes in traits across geographical areas, illustrating how populations adapt to different environments. Moving on, we see the patterns of speciation, mainly anagenesis and cladogenesis. Anagenesis is when a single species gradually transforms into a new form, whereas cladogenesis involves a branching event, creating new species from a common ancestor. When discussing modes of speciation, allopatric speciation occurs when populations are geographically separated, leading to divergence. In contrast, sympatric speciation happens without physical separation, often driven by other factors like ecological niches or behavioral differences. The tempo of speciation can be gradualistic, where changes accumulate slowly over time, or punctuated, with rapid shifts followed by long periods of stability. Reproductive isolating mechanisms are critical here, as they prevent gene flow between diverging populations, maintaining their distinct identities. Secondary contact and reinforcement are also key concepts. When populations that have diverged come back into contact, reinforcement can strengthen reproductive barriers, reducing the chances of hybridization. Speaking of hybrids, hybrid zones are regions where two species interbreed, often resulting in introgression or gene flow between species, which can complicate the boundaries we define as species. Finally, understanding different species concepts helps us interpret these processes, whether it's the biological, morphological, or phylogenetic perspective. All these mechanisms and concepts work together to shape the incredible diversity of life we see today. Let's talk about the mitochondrial DNA, or mtDNA, in these warblers. All individuals in this group have Townsend's mtDNA, which is a key genetic marker. However, some individuals also carry Hermit mtDNA, while others only have Townsend's. This variation is important because it helps us understand hybridization patterns. Interestingly, all the individuals we see here have Hermit mtDNA, which suggests that the hermit warbler's mtDNA is quite prevalent in this population. We also see different types of warblers: the hermit warbler, Townsend's warbler, and hybrids that are a mix of both species. These hybrids tend to show intermediate characteristics, meaning they look and behave somewhere in between the two parent species. A crucial point is that hybrids inherit their mtDNA from their mothers, which means the mitochondrial DNA sequence is species-specific and passed down maternally. So, by analyzing the mtDNA, we can trace back the maternal lineage and better understand the hybridization dynamics in this region, especially across the Pacific Ocean. Overall, this genetic information helps us piece together how these species interact and evolve in their natural habitats. In this slide, we're looking at gene regions that are similar across different Helianthus species. Specifically, we see regions similar to H. annus and H. petiolaris. These similarities help us understand the genetic relationships and how these species might have evolved from common ancestors. The DNA comparison of the S region of the genome is particularly interesting. Here, we compare H. anomalus, which is a naturally occurring species, with an experimental hybrid that results from crossing H. annus and H. petiolaris. It's worth noting that not all parts of this region were tagged in the analysis, which is an important consideration when interpreting the data. Ultimately, this comparison sheds light on the genetic makeup of these species, showing us how hybridization can influence genome structure and variation, tying back to our overall theme of genetic diversity and evolution in sunflowers. So, what exactly is population ecology? Well, it's the study of how members of a population interact with their environment. Basically, it looks at how individuals within a species relate to the natural world around them. This includes understanding how they find resources like food and water, how they compete or cooperate with each other, and how environmental factors influence their survival and reproduction. These interactions are crucial because they help us understand the dynamics of populations over time. By examining these relationships, we gain insights into the broader ecological processes that shape ecosystems. This understanding ties into the overall theme of how living organisms adapt and thrive within their environments. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's talk about density and dispersion, two important concepts in understanding populations. Density refers to the number of individuals in a given area or volume, so it helps us understand how crowded or sparse a population might be. For example, a high density means many individuals are packed into a small space, while a low density indicates they're more spread out. Dispersion, on the other hand, is all about the pattern of spacing among those individuals within the population boundaries. It shows us how individuals are distributed, whether they're clumped together, evenly spaced, or randomly scattered. Both density and dispersion are crucial because they influence how populations interact with their environment and with each other. They give us insights into behaviors like competition, resource use, and mating. Understanding these patterns helps us see the bigger picture of how populations survive and thrive in their habitats. Density, or the idea of how many individuals are in a given area, is a really dynamic concept. It's something we can determine, but it's often quite challenging to do so accurately. In most cases, trying to count every single individual in a natural population isn't practical or even feasible. Think about a dense forest or a large ocean habitat. Trying to get an exact count there would be nearly impossible. Instead, ecologists use various methods to estimate population density, which helps us understand how populations are distributed and how they might be changing over time. So, while it's a complex task, understanding density is crucial because it ties directly into how populations grow, compete, and interact with their environment. Density is essentially the result of a dynamic interplay between processes that add individuals to a population and those that remove them. On the one hand, births and immigration work to increase the number of individuals in a population. Births are the natural increase through reproduction, while immigration involves individuals moving into the population from elsewhere. On the other hand, we have processes like emigration and deaths that reduce the population size. Emigration is when individuals leave the population to go elsewhere, and deaths obviously decrease the number of individuals. So when we think about population density, it's really about the balance between these added and removed individuals. If births and immigration outpace deaths and emigration, the population density will increase. Conversely, if more individuals leave or die than are added, the density will decrease. This balance is fundamental to understanding how populations grow, shrink, or stabilize over time, which ties back to the larger themes of population dynamics and ecological interactions we've been discussing. Let's take a closer look at demography. Demography is essentially the study of the vital statistics of a population, things like birth rates and death rates, and how these numbers change over time. These changes are really important because they help us understand population growth or decline and how societies evolve. In particular, demographers pay close attention to birth rates and death rates. Birth rates tell us how many people are being born in a given period, while death rates indicate how many people are passing away. Monitoring these rates gives us insight into the health, stability, and future trends of a population. Understanding these key factors is crucial because they connect directly to the overall theme of population dynamics. By analyzing these trends, we can better plan for resources, healthcare, and policy needs. So, demography isn't just about numbers. It's about understanding the story of a population's growth and change over time. Let's take a closer look at the factors that influence population changes. First, we have natality, which refers to the birth rate, and it's closely related to fecundity, or the biological capacity to reproduce, and fertility, which is the actual number of offspring produced. These elements are fundamental because they determine how a population grows or shrinks over time. Next, immigration plays a significant role. When people move into a region, they increase the population, while emigration, or people leaving an area, can cause it to decline. Both are crucial in shaping demographic patterns. Mortality and survivorship are also key factors. Mortality rates reflect the number of deaths in a population, and survivorship indicates the likelihood of individuals surviving through different age stages. Together, they influence the population's age structure and longevity. Speaking of age structure, it's essentially the distribution of different age groups within a population. This distribution affects future growth prospects and economic development. All these factors, births, deaths, immigration, emigration, and age structure, are interconnected and help us understand the dynamic nature of populations. Recognizing these influences allows us to better grasp how populations evolve over time and their implications for society and ecosystems. Let's take a closer look at life tables. A life table is essentially an age-specific summary of how a population survives over time. It provides a detailed picture of survival patterns at different ages. The most effective way to construct a life table is by following the fate of a cohort, which is a group of individuals born around the same time. By tracking this group throughout their lives, we can gather accurate data on survival rates and mortality at each age. This approach helps us understand the dynamics of population survival and is fundamental for demographic studies and understanding population health. Connecting this to our overall theme, life tables are crucial tools for analyzing population trends and planning for future needs. Let's take a closer look at patterns of dispersion. As we've discussed, environmental and social factors play a crucial role here. These factors influence how individuals are spaced within a population. For example, resources like water, food, or shelter can cause animals to cluster together or spread apart. Social behaviors, such as territoriality or mating habits, also impact how populations are arranged in space. Recognizing these patterns helps us understand the broader dynamics of populations and how they adapt to their environment. So, by examining these influences, we gain a better picture of how living things organize themselves in their habitats. Let's take a closer look at the appearance of different dispersions. As you can see, dispersions can vary quite a bit in how they look and behave. The key point here is that the appearance depends largely on the size of the dispersed particles and how they interact with the medium. For example, in a colloidal dispersion, the particles are small enough that they stay suspended, giving it a cloudy or opaque look. On the other hand, in a solution, the particles are so tiny that the mixture appears clear and transparent. Understanding these visual differences helps us identify and classify dispersions, which is essential for many applications across industries. So, recognizing how dispersions look under different conditions ties back to our overall theme of understanding their properties and behavior. Clumped dispersion is when individuals tend to gather in specific patches or groups rather than being spread out evenly. This pattern often occurs because resources like food, water, or shelter are unevenly distributed across the environment. When these resources are concentrated in certain areas, animals or plants naturally cluster around them to maximize their survival chances. It's also common in social species, where group living provides protection or helps with reproduction. Recognizing clumped dispersion helps us understand how organisms adapt to their surroundings and how their populations are structured. It's a key concept because it influences things like resource management and conservation strategies, which are essential themes in ecology. So, understanding why and how organisms cluster together gives us a deeper insight into their behaviors and their interactions with the environment. A clumped dispersion is when individuals tend to gather in patches rather than being evenly spread out. This pattern often reflects the way resources are distributed in the environment or specific behaviors of the species. For example, many animals, like these wolves, live in groups because it boosts their chances of success. Living together makes hunting more effective, as they can coordinate and take down larger prey. It also helps spread out the responsibilities of protecting and caring for their young, which is crucial for their survival. Plus, grouping together helps them defend their territory by excluding other individuals. So, this clumped pattern is really about maximizing survival and reproductive success based on resource availability and social behavior. It ties into the overall theme of how organisms adapt their spatial arrangements to improve their chances in the wild. By contrast, uniform dispersion is when individuals are evenly spaced. This means that instead of clustering or being randomly scattered, each individual maintains a consistent distance from the others. It's like if you imagine a field where plants are spaced out so that none are too close or too far apart. This kind of distribution is often seen in situations where resources are evenly distributed or where social interactions require a certain amount of personal space. Recognizing uniform dispersion helps us understand how populations organize themselves in environments where competition or territorial behavior is a factor. So, in the broader context of our presentation, identifying uniform dispersion can give us insights into the underlying social or environmental influences shaping these patterns. A uniform dispersion is a pattern where individuals are evenly distributed across an area. This means that each individual maintains a certain distance from others, leading to a regular spacing. Such arrangements can often be influenced by social interactions, especially territoriality. For example, animals may establish and defend territories to prevent others from encroaching. Taking a specific example, think about birds nesting on small islands. These birds, like king penguins on South Georgia Island in the South Atlantic Ocean, often show this uniform spacing. It's maintained by aggressive interactions between neighbors, which helps keep everyone in their own space. Understanding these patterns helps us see how social behavior and competition shape the distribution of species in their environments. It's a key part of understanding population dynamics and how animals interact within their habitats. In random dispersion, which is covered in section 426, the key idea is that the position of each individual is independent of the others. This means that where one individual is located doesn't influence where the next one might be. It's like tossing seeds randomly across a field without any pattern or clustering. Now, an important point to remember here is that the overall dispersion, the way individuals are spread out across an area, actually depends on how resources are distributed. If resources are evenly spread, individuals might be more evenly dispersed as well. Conversely, if resources are clumped in certain areas, you might see some randomness in individual positions, but overall, their distribution still reflects where resources are available. This concept ties into our broader discussion of how organisms or particles distribute themselves in space and what factors influence that pattern. Random dispersion is just one way, and understanding resource distribution helps us make sense of why these patterns emerge. A random dispersion is a pattern where the position of each individual is independent of others. In other words, one person's placement doesn't influence where someone else is located. It's a completely chance-based arrangement. For example, think about dandelions growing from wind-blown seeds. The seeds land at random, without any specific pattern, and then later germinate. This randomness in seed landing illustrates how individuals can be scattered in a truly unpredictable way. Understanding this helps us see how some populations or organisms distribute themselves naturally, often influenced by factors like wind or other environmental forces. It ties into the broader theme of how different dispersal mechanisms shape the spatial distribution of species in their habitats. Today, we're going to look at population growth models. Specifically, we'll focus on two main models that help us understand how populations expand over time: the exponential growth model and the logistic growth model. These models are fundamental because they give us a framework to analyze and predict population dynamics. The exponential growth model describes how populations can grow rapidly when resources are unlimited, leading to a J-shaped curve. On the other hand, the logistic growth model takes into account environmental limitations, showing how growth slows as the population approaches the carrying capacity of its environment. Understanding these models is crucial because they connect directly to the overall theme of how populations interact with their ecosystems and resources. By comparing these models, we can better grasp the factors that influence population stability and change. Let's take a closer look at how mathematical models help us understand population growth. Essentially, population numbers change due to two main factors: natality, which is the birth rate, and mortality, the death rate. On this slide, you see the basic equation. Delton divided by deltat equals b minus d here. Delton represents the change in population size over a certain time period, deltat. The variable b is the natality rate, and d is the mortality rate. Now, if we simplify this, we define a growth rate, r, which is simply the difference between birth and death rates. R equals b minus d. This growth rate is crucial because it tells us whether the population is growing, stable, or declining. When r equals 0, it indicates a stable population. Births exactly balance deaths. If r is greater than 0, the population is increasing in size. Conversely, if r is less than 0, the population is decreasing. These relationships can also be expressed in a differential equation. dn divided by dt equals r times n, where n is the current population size. This equation models how the population changes over time based on its current size and growth rate. Understanding these models helps us grasp the dynamics of populations, whether we're studying conservation, resource management, or ecological balance. It's a fundamental piece of the larger picture of how populations grow and change in natural systems. Dispersal can have a significant impact on the growth rate of some populations. When we talk about dispersal, we're referring to the movement of individuals from one population to another. This movement can be in the form of immigration, which is when individuals enter a population, or emigration, when they leave. Immigration, represented by the letter I, increases the number of individuals in a population, while emigration, represented by E, decreases it. These flows of individuals directly influence the population's growth rate. Now, the per capita growth rate, think of it as the average growth per individual, can be calculated by taking the birth rate, subtracting the death rate, and then adding the net migration rate, which is immigration minus emigration. So mathematically, it's expressed as R equals B minus D plus I minus E. This equation shows us that dispersal isn't just about movement. It actually plays a key role in how fast or slow a population grows, especially in connected or dynamic environments. Understanding this helps us see how populations are not isolated. They're constantly influenced by movement in and out, shaping their growth patterns over time. Each population has a characteristic intrinsic rate of increase, which is really the maximum rate at which that population can grow under ideal conditions. It's important to understand that this rate is influenced by the species' life history characteristics, which essentially determine its biotic potential. Key factors include the age at which individuals start reproducing and how much of their lifespan they spend being able to reproduce. For example, some species begin reproducing early in life, while others start much later. Then, we look at how many offspring they produce during each reproductive period. Small organisms, like bacteria, tend to have the highest intrinsic rates of increase because they reproduce quickly and in large numbers. In contrast, larger animals such as elephants have much lower intrinsic rates because their reproduction is slower and they produce fewer offspring over their lifetime. Understanding these differences helps us see how various species adapt their reproductive strategies to their environments and how their life history traits influence their potential for population growth. The exponential growth model describes the rate of expansion of a population under ideal, unregulated conditions. What this means is that, in a perfect environment with unlimited resources, a population can grow rapidly without any constraints. The model essentially captures how populations could increase if nothing is holding them back. When we talk about the ideal of an unlimited environment, we're imagining a scenario where resources like food, space, and other necessities are abundant, so the population isn't limited by these factors. This concept helps us understand the potential for rapid growth, but it's also a reminder that in real-world situations, such ideal conditions are rare. Throughout this presentation, we'll see how this model provides a foundation for understanding population dynamics, especially when examining what happens when growth is unchecked. Let's talk about exponential population growth. Typically, the larger the population, the faster it tends to grow. When we plot the number of organisms over time, especially under optimal conditions, we usually see this distinctive exponential growth pattern. This means the population increases at a rate proportional to its current size, leading to a rapid and accelerating rise. Recognizing this pattern is important because it highlights how populations can expand quickly when resources are abundant and conditions are ideal. This concept ties into the broader themes of population dynamics and how environmental factors influence growth rates. This slide doesn't contain enough information. That's why we're moving to the next slide. A key feature of the exponential growth model is that the rate at which a population grows depends on the number of individuals already in the population. In other words, the larger the population gets, the faster it grows. This creates a kind of feedback loop where growth accelerates as more individuals are added. It's important to understand that this isn't a linear process. Growth isn't just adding a fixed amount each time. It's increasing proportionally to the current population size. This concept helps us see how populations can expand rapidly under ideal conditions. Throughout this presentation, we're exploring how these models illustrate real-world growth patterns, and this feature, the dependence on current population size, is central to understanding exponential growth's power and potential impact. No population can increase exponentially forever. While exponential growth might happen for a certain period, eventually, limiting factors come into play and slow things down. These limiting factors are what we call environmental resistance. They prevent populations from growing endlessly because resources like food, water, and space become scarce as the population increases. As the number of individuals, or N, gets larger, this environmental resistance also increases. Now, K, or the carrying capacity, is the maximum population size that an environment can support indefinitely. But it's important to remember that in real life, K isn't a fixed number. It can change over time as environmental conditions shift. This means that populations are always responding to their environment, and their growth isn't just a simple, endless curve. Instead, it's a dynamic process influenced by the available resources and environmental factors. Let's take a closer look at the logistic growth model and how it reflects the reality of limited environments. In nature, a population might initially grow very quickly, almost exponentially, when resources are abundant. However, this rapid growth can't continue forever. Eventually, environmental factors such as food availability, space, or other resources will start to limit that growth. This means the population growth rate slows down as it approaches a maximum capacity, often called the carrying capacity. Understanding this helps us see that populations don't grow unchecked forever. Instead, they are constrained by their environment. This concept ties back into our overall theme of how populations interact with their ecosystems and the factors that regulate their growth. Let's take a closer look at the logistic equation, which helps us understand how populations grow when environmental resistance comes into play. This growth pattern often creates that characteristic S-shaped curve we've discussed earlier. The key idea here is that growth is not unlimited. Instead, it's regulated by the environment, leading to a slowing down as the population nears its carrying capacity, often denoted as K. The logistic equation itself is a bit more refined than the simple exponential growth model. It looks like this. dn/dt equals r times n multiplied by the term k minus n divided by k. When the population size n is small, the term kn/k is close to 1. That means growth isn't heavily constrained, so the population can expand rapidly. But as n gets larger and approaches k, this term approaches 0, which significantly limits growth. It's like hitting a ceiling. Growth slows down and eventually stabilizes. However, it's important to note that populations don't always just gently settle at k sometimes. They overshoot it, becoming larger than the carrying capacity, which can lead to a population crash afterward. This dynamic is crucial because it shows how populations can fluctuate around their environmental limits, and understanding this helps us grasp how ecosystems maintain balance or experience instability over time. The logistic growth model describes how a population grows when it faces certain limitations. Unlike exponential growth, where the population keeps increasing rapidly, this model considers factors that slow down that growth as the population gets larger. These limiting factors could be resources like food, space, or even other environmental constraints. What's interesting is that this model helps us understand real-world populations more accurately because it reflects how growth actually happens in nature. As the population approaches its maximum capacity, or what we call the carrying capacity, growth slows down and eventually stabilizes. This concept is really important because it connects to the overall theme of how populations interact with their environment and the limits that shape their growth over time. Today, I want to walk you through the 3 key phases that are often observed in various growth or development processes. First, we have the exponential phase, which is characterized by rapid, often accelerating growth. This is when things start to pick up quickly, and progress can seem almost unstoppable. It's a critical period because it sets the foundation for what's to come. Next, we move into the plateau phase. This is when growth begins to level off. It might feel like progress has stalled, but in reality, it's a natural part of the process. During this stage, systems are often consolidating and stabilizing. Recognizing this phase helps us understand that not every period of stagnation is negative. Sometimes, it's just a necessary pause before moving forward again. Finally, there's the transitional phase. This is the phase where change starts to happen once more, leading to another cycle of growth or development. It's like a bridge that connects the plateau to renewed exponential growth. Understanding these phases helps us see the bigger picture of how progress unfolds over time, especially in complex systems or projects. Throughout our overall theme, recognizing these phases enables us to better manage expectations and strategies, ensuring we're prepared for each stage and can adapt accordingly. Now let's take a closer look at the logistic curve, which is often called the S-shaped curve. This type of curve is really important because it models how many processes grow over time in a way that's realistic for many real-world situations. At the beginning, you'll notice the growth is slow. This is because of initial barriers or limited resources. Then, as conditions become more favorable, the growth accelerates rapidly, creating that steep middle section of the S-shape. But what's really interesting is what happens next. As resources become limited or competition kicks in, the growth starts to slow down and eventually levels off. That's the upper plateau, where the system reaches a sort of equilibrium. This S-shape elegantly captures the idea of growth that starts slow, speeds up, and then stabilizes, something we see in population dynamics, technology adoption, and even in business growth. Understanding this curve helps us see the natural limits of growth and the importance of timing and resource management in strategic planning. It ties into our overall theme of modeling complex systems and predicting how they evolve over time. How well does the logistic model fit the growth of real populations? Well, in many cases, especially with laboratory populations of certain animals, the growth patterns actually align quite well with the S-shaped, or sigmoid, curves that the logistic model predicts. This means that early on, the population grows slowly, then speeds up as resources become more available, and finally slows down again as it approaches the environment's carrying capacity. It's important to highlight that while the logistic model captures this general trend, real populations can sometimes deviate due to factors like environmental changes or species interactions. Overall, this fit suggests that the logistic model is a useful tool for understanding and predicting population growth in controlled settings, and it helps us connect the mathematical concepts we've been discussing to actual biological systems. Some of the assumptions built into the logistic model do not apply to all populations. It's important to remember that while the logistic model provides a useful framework, it's a simplified representation of reality. It assumes, for example, that growth rates are constant and that resources become limiting in a predictable way. However, in real-world populations, factors like environmental variability, social behaviors, and unique ecological conditions can cause deviations from the model's predictions. That's why I like to emphasize that the logistic model serves as a baseline or a point of comparison. It gives us a foundation to understand and analyze how populations grow, but we always need to compare it with actual data. This way, we can identify where real populations diverge from the model and gain deeper insights into the underlying causes. In the context of our overall theme, this highlights the importance of not relying solely on models, but using them as tools to guide our understanding and decision-making. They're valuable, but they're just one piece of the bigger picture when studying population dynamics. Let's take a closer look at the logistic population growth model and how it relates to different life histories. This model helps us understand that populations don't always grow at the same rate, especially as they approach their carrying capacity. The key idea here is that resource availability isn't constant. It depends on the specific situation and environmental conditions. Because of this, natural selection may favor different traits, depending on factors like population density and the environment. For example, in K selection, organisms tend to live and reproduce near the carrying capacity. They are quite sensitive to changes in population density, which helps maintain stability. On the other hand, our selection is characterized by organisms that reproduce rapidly, especially in unpredictable or variable environments where populations often fluctuate well below K. Understanding these strategies helps us see how different species adapt to their environments and optimize their survival and reproduction. This ties into our broader theme of how life histories and environmental factors shape population dynamics. Let's take a closer look at how the logistic growth model compares to the exponential growth model. On this slide, you'll see the term carrying capacity. That's a key concept in the logistic model. It represents the maximum population size that an environment can sustain over time. Now, with exponential growth, the population increases rapidly without any regard for this limit. It's like an unchecked explosion. The population keeps doubling, tripling, and growing faster and faster, no boundaries in sight. But in reality, resources are limited, And that's where the logistic growth model comes in. It incorporates the idea of carrying capacity, which slows down growth as the population approaches that limit. So, instead of an endless rise, the growth curve levels off, creating an S-shaped or sigmoid curve. Understanding these differences is crucial because it helps us predict how populations grow in real-world environments, balancing between ideal conditions and resource constraints. This comparison ties directly into the overall theme of understanding population dynamics and the factors that influence growth over time. Let's talk about carrying capacity. This is the maximum number of individuals that an environment can support without the population growing or shrinking. In other words, it's the point where the environment can just sustain the population as it is, no net increase, no net decrease. Now, an important concept related to this is the balance between death rates and birth rates. When these rates are equal, the population size remains stable. So, a stable population occurs when the number of people being born equals the number of people passing away. Understanding carrying capacity, and this balance helps us see how populations grow, stabilize, or decline over time, which fits into our broader discussion of population dynamics and how organisms interact with their environments. Let's talk about metapopulations, which are essentially collections of several local populations. Many species are not spread out as one big, continuous population across the landscape. Instead, they consist of a series of smaller, distinct populations that are distributed across different habitat patches. This patchiness often happens because these species have narrow physiological requirements, meaning they need very specific conditions to survive. As a result, their populations tend to be small and isolated, occurring only in suitable patches of habitat. When these small populations are connected through dispersal, that is, individuals moving from one patch to another, we refer to the entire system as a metapopulation. Now, within this framework, we distinguish between good habitats called sources and less suitable ones called sinks. Sources are areas where local reproductive success exceeds local mortality, so these patches produce more individuals than are lost. On the other hand, sinks are areas where local reproductive success is less than local mortality, meaning these patches rely on individuals dispersing from sources to maintain their populations. Understanding these concepts helps us see how populations are maintained across fragmented landscapes and highlights the importance of habitat quality and connectivity for species survival. Let's take a closer look at the concept of metapopulation structure. This idea is fundamental in understanding how populations are organized across different habitats or regions. Essentially, it refers to a group of spatially separated populations of the same species that interact through processes like migration and dispersal. The key point here is that these populations are connected, but they also function somewhat independently. This structure allows for resilience because if one local population declines or even goes extinct, others can recolonize the area. Understanding this structure helps us grasp how species persist in fragmented environments, which is increasingly relevant in conservation biology. It highlights the importance of maintaining connectivity between populations to support overall species survival. So, in the context of our overall theme, recognizing the metapopulation structure emphasizes the need to consider not just individual populations, but the broader landscape and how these populations interact as a network. This interconnectedness is crucial for effective management and conservation strategies. Today, I want to talk about forest metapopulations. Many forests are actually arranged as metapopulations, which means they consist of separate patches or groups of trees that are interconnected in some way. Different species of trees are dispersed from each other across these patches, creating a mosaic of forest areas. This natural arrangement happens quite often, especially in diverse ecosystems where various species occupy different niches and habitats. However, it's important to recognize that this natural pattern is increasingly impacted by human activities. For example, when humans log forests to create agricultural land or develop towns and cities, they fragment these forest patches further. This fragmentation can disrupt the connectivity between tree populations, making it harder for species to survive, reproduce, and maintain genetic diversity. So, understanding forest metapopulations helps us see how natural processes and human actions influence forest health and resilience. This concept ties into the broader theme of conservation and sustainable management, emphasizing the importance of maintaining connectivity in forest ecosystems to support biodiversity. Now, let's talk about naturally occurring metapopulations. These are groups of populations that exist across different habitats or locations, but are connected through the movement of individuals. Essentially, they form a network where local populations can go extinct, but can also be recolonized by individuals from neighboring populations. This dynamic helps maintain the overall stability of the species in a given area. It's a fascinating concept because it highlights how species can persist despite local setbacks, thanks to these natural dispersal and recolonization processes. Understanding these patterns is crucial because it emphasizes the importance of habitat connectivity and landscape-level conservation. It reminds us that protecting just one patch of habitat isn't enough. It's about maintaining the larger network that allows these populations to thrive and bounce back over time. Let's talk about population selection. When we're choosing the populations for our study, it's crucial to be deliberate and systematic. We want to ensure that the populations we select truly represent the broader group we're interested in. This means considering factors like demographic diversity, geographic location, and relevant characteristics that could influence our results. By carefully selecting our populations, we help improve the validity and generalizability of our findings. It's also important to be transparent about our selection process, so others can understand and replicate our approach if needed. Overall, thoughtful population selection is a foundational step that sets the stage for meaningful and reliable results throughout the entire research process. When we talk about fitness in the context of evolution, we're really referring to reproductive success. And this is a crucial concept because natural selection, which is the main driver of evolutionary change, operates through differences in this reproductive success. Essentially, some genotypes are more successful at reproducing than others, and that leads to changes in the frequencies of these genotypes within a population over time. So, when we say that natural selection is causing evolution, we're highlighting that it's the unequal reproduction of different genotypes, some producing more offspring than others, that causes the shift in genetic makeup. This differential success, or fitness, determines which traits become more common in future generations. Understanding fitness helps us see how populations adapt to their environments and evolve over generations, tying directly into the broader theme of how life changes and adapts through natural processes. So, here we see the concept of natural selection, which was discovered independently by Charles Darwin in 1837 and Alfred Russel Wallace in 1858. This idea really revolutionized how we understand the evolution of species. Darwin's seminal work, The Origin of Species, was published in 1859 and laid out this groundbreaking theory in detail. Natural selection explains how species adapt over time through a process where the individuals with advantageous traits are more likely to survive and reproduce. It's such a powerful concept because it provides a natural explanation for the diversity of life we see around us, without needing to invoke any supernatural ideas. And it's interesting to note how some of the greatest minds of the time viewed this discovery. T.H. Huxley famously remarked, "How incredibly stupid not to have thought of that," highlighting just how obvious and elegant this idea is once you understand it. This discovery is a cornerstone of modern biology, connecting deeply to the overall theme of evolution and the dynamic nature of life. It's a perfect example of how scientific insights can profoundly alter our understanding of the natural world. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide.